Okay, welcome to Let's Get to the Marks. We're looking at carbohydrates for the SNAB Salters Biology A-level. Which of the following describes lactose? So we've got disaccharide, modified disaccharide, monosaccharide, or protein. Well, the answer is disaccharide. Hopefully you all know that. Lactose is a disaccharide made of two monomers, two monosaccharide sugars. And lactose is made of glucose and fructose. Okay, so as I go through this walkthrough on carbohydrates for SNAP, make sure you pause the video and have a go at the questions. Okay, so moving on. Starch is an important component of the human diet. The main sources of starch are plants such as maize and potatoes. Starch is composed of amylose and amylopectin. Which of the following terms is the correct description of amylose? A disaccharide, monosaccharide, polysaccharide, or trisaccharide? Make sure you pause and have a go. All right, let's go through the answers. So, of course, starch is made of amylose and amylopectin, and these are both polysaccharides. They're made up of many, many monomers of glucose. They're energy storage molecules, and starch is the energy storage molecule that you'll find in plants. It's what they do with the glucose made from photosynthesis, or one of the things they do. And amylose is a coiled, unbranched chain of starch. Starch can be broken down by a condensation reaction or a hydrolysis reaction. So which one is a breaking reaction? Hydrolysis, the word lysis, L-Y-S-I-S, that means breaking. So hydrolysis, or hydrolysis, however you want to say it, is the reaction for breaking a polymer into monomers. So breaking a big molecule into small molecules. Doesn't matter whether we're dealing with proteins, fats, or carbohydrates, or DNA, it's the same breaking molecule in biology. The answer here is D, because carbohydrates contain glycosidic bonds, lipids contain ester bonds. So the correct answer is D. Okay, next question. Give one structural difference between amylose and amylopectin. So what I'm doing with my grade D GCSE art is drawing you a molecule of amylopectin. You can see my amylose above, that's the coiled one. So you can see amylopectin made of the exact same monomers, these little crusty circles I've drawn, but there's a difference in the structure. Amylopectin is highly branched, so it has 1 to 6 branches, 1 to 4 glycosidic links and 1 to 6, which of course means it's very quick to hydrolyze and release energy. Amylose is a better long-term storage molecule. It's a coiled chain. It's got like an alpha coil, the way it's like coiled like a spring, but it's actually a straight chain with no branches. Okay, glycogen and starch can be hydrolyzed by enzymes. Which row shows the correct features of the structure of glycogen? Well, glycogen is very much like amylopectin, very similar. Amylopectin is found in plants. Glycogen is the energy storage molecule in animals, and glycogen is highly branched. So if you're highly branched, what must be your glycosidic links? 1 to 4 are the ones which just join two monomers together, and then 1 to 6 is where you go vertically, or you go vertically up or down to another branch. So we're looking here at C, because it has 1 to 4 and 1 to 6 glycosidic bonds, and it's branched. You can't have 1 to 4 and 1 to 6 glycosidic bonds and be unbranched. So D is definitely incorrect. And if you're branched, you're going to have more than one type of bonding. And it's those lots and lots of branches in glycogen that actually give it one of its important features as a storage molecule, which is that it's hydrolyzed easily. Okay, here we go. Explain how the structure of glycogen allows it to be an energy store. So what are some of the funky things about glycogen that make it good as a store of energy? Right, number one, it's got lots and lots and lots of bonds, covalent bonds, all containing lots of energy. So when you hydrolyze those, you release that energy. So it's exothermic. You're going to release that energy when you break those glycosidic bonds down. Okay, and glucose has lots of bonds. So when you break up that glucose in respiration, you're going to release lots of ATP, or produce lots of ATP, or release lots of energy. It's highly branched. What this means is that it's really, I wouldn't say it's easy to hydrolyze, but it's quick to hydrolyze. It's much faster to hydrolyze a branch chain molecule than a straight chain molecule. So you can get access to that energy more quickly. And of course, it's made of alpha glucose. Glucose is the molecule required for respiration. So that's important. And one of the most important things that people forget about with all of these polymers, all of these polysaccharides, they're insoluble. So amylose, amylopectin, and glycogen in animals are insoluble. And that's important. It doesn't have an osmotic effect. So what do we mean by osmotic effect? If you stored just glucose molecules in a cell, it would lower the water potential, cause water to move in by osmosis. The cell would swell up and it could damage the organelles or burst the cell or the organelles. So easy to describe that by saying it's insoluble, so no osmotic effect. All right, this next question. Maltose and trehalose are disaccharides. Trehalose is formed from two molecules of alpha glucose. Here's a diagram of the molecule of trehalose. Describe the reaction that joins two alpha glucose molecules together to form a disaccharide. So it's not specific to this. What's the name of the reaction that joins two molecules together? Pause the video, have a go. Okay, hopefully you know the name of the reaction. It's going to form this glycosidic bond here, and it's going to join these two molecules together. If we were dealing with lipids, it would form an ester bond. If we were dealing with proteins, it would form a peptide bond. And of course, it's the condensation reaction where water is removed, or water is released or removed, and you get that bond formed between two biological molecules. And in this case, it's the forming of a glycosidic bond between these two glucose molecules. Okay, so really the marks here are for describing the reaction. So it's a condensation reaction, water is produced, glycosidic bond is made. They're the key details there. Right, let's move on. Maltose and trehalose, yet again, we meet again. Trehalose is formed from two molecules of alpha glucose. Here's the diagram. Compare and contrast the structures of trehalose and maltose. Well, they're both made. What do we mean by compare and contrast, first of all? So I was just about to say they both contain alpha glucose, but let's go over what do we mean by compare and contrast. So compare in SNAB, A-level biology, when you see the word compare, it means what's the same between these two things. And contrast means what is different. So what we want to do here is say two things that are the same, or one thing that's the same and two things that are different, whatever. Best to say three things, 2 marks, but lots of things on the mark scheme. So both are made of alpha glucose monomers. So that's a similarity. What type of bond do they both have? What type of bond joins the monomers together? You guessed it. They're both carbohydrates, so it's the glycosidic bond. So both contain glycosidic bonds. So we can get a mark for comparing by saying, look, they're both alpha glucose, made of alpha glucose monomers, and both contain glycosidic bonds. However, what's different about them? I don't know if you've noticed, but the second molecule of alpha glucose in this trehalose is upside down. And look where the oxygen atom is. It's reversed or inverted. So in trehalose, one of the glucose molecules is upside down or reversed 180 degrees. So that's one of the differences. Now, you would definitely have both the marks here because we've said three things here, two similarities and one difference. What's some other differences? Well, there's some other differences in the. Okay, one of the other differences when we look here is that if you look here at this oxygen atom I'm highlighting, this is how we number a glucose molecule. We start with the oxygen atom and we go around 1, 2, 3, 4, 5. 6 is the one that's like branching off the top of the glucose molecule. Look at the reversed one. I'm going around this way. 1, 2 from the oxygen, 3, 4, 5, and then 6 on the inside there. That means that the link here in trehalose is a 1-to-1 glycosidic link. Hopefully you remember the link in a normal maltose molecule, and that's going to be 1-to-4 glycosidic link. So trehalose is a glycosidic 1-1-1 link or bond, glycosidic bond of 1-1, and maltose has a 1-to-4 glycosidic link. So that's the difference when you invert one of the molecules. All right, let's move on. Okay, next question. Bada bada bing, zing zing. Explain how the structures of amylopectin and glycogen make them suitable for storing energy. I mean, we kind of just looked at this really. Both of them are highly branched. So amylopectin you'll find in plants. You don't find it in animals. Glycogen you find in the liver and muscle cells of animals. You also find glycogen in fungi as well. So they're both compact and you can fit a large mass in a small volume, although often that's not on the mark scheme in SNAB. talking about the compact nature of the energy storage molecules, but they're both made of glucose monomers. And of course, why is that important? Well, glucose is the molecule we use for respiration, or living things generally use for respiration. There's lots of covalent bonds in a glucose molecule, which means that when they're broken, it will release a lot of energy. So contains lots of bonds, lots of energy. One of the really key factors with amylopectin and glycogen, which is why they've mentioned them, is that they are highly branched. They have lots of branches. They have 1 to 4, 1 to 6 glycosidic links. You could write that if you want, instead of saying they're highly branched. And this means they can be hydrolyzed, broken down quickly to release energy or to release glucose. And of course, also the final point, which we've made before, they're insoluble, so they don't have an osmotic effect. Okay, moving on. Maltose and trailer-hose are disaccharides. Yet again, trailer-hose and maltose making a comeback. Some flying insects store trailer-hose and glycogen in their wing muscles. Trailer-hose is broken down to glucose by the enzyme trailer-hase. Which of the following describes how glucose is produced from trailer-hose? And of course, we know that one is hydrolysis. So if you're going to break down a molecule to release glucose, it's going to be hydrolysis. Okay, let's move on. Insect flight uses a lot of energy. Explain the advantage of insects storing both trailer hose and glycogen. So first up, glycogen is a polymer, means it's made of many glucose molecules. Trailer hose is a disaccharide, so just 2 glucose molecules. So what's the advantage of that? Well, first of all, why is it an advantage to store lots of glycogen? Um, Glycogen, of course, we know contains many glucose molecules, so you can store lots of energy. It's compact in a small space. We can also say that glucose is what's used for respiration, to produce ATP or to release energy. And so that's why it's useful to have glycogen as a storage molecule. We can also point out that glycogen is highly branched, which we've mentioned many times, which is great for quick energy release, or it can be readily hydrolyzed, or for rapid energy release or rapid production of ATP. Um, Really, it releases glucose that's then used in respiration, but you get the idea. It's quick to access. Glycogen's also insoluble, so it doesn't have an osmotic effect, so that's really key. You don't want your your energy storage to become a problem where it's making osmosis occur in places you don't want. Now, the advantage of trailer hose, okay, it does have an osmotic effect. It might be soluble, but the advantage of trailer hose is that it only contains 1 glycosidic bond. So it's even faster to access those glucose molecules for energy. So if I'm a little flyer and I need to make a quick divert move and duck out the way of someone trying to swap me with a hand, I can tuck into that trailer hose and use that to get the energy. Okay. Um, there you go. So that should get us our 4 marks. There's really 3 points to talk about glycogen here. The advantage of the trailer hose is that it can be quickly broken down to provide glucose for respiration, as it only contains 1 glycosidic bond. Monosaccharides join to form disaccharides and polysaccharides. State the type of reaction where 2 monosaccharides join to form a disaccharide. What do we call the reaction where they both join together? That's going to be condensation. All bond making is condensation, and bond breaking is hydrolysis with biological molecules. And then down here, of course, what type of bond gets formed? It's a glycosidic bond. Esters are found in fats, peptides in proteins, hydrogens in the tertiary structure of protein, and of course, hydrogen bonds hold together water molecules and the 2 strands in DNA. Monosaccharides join to form disaccharides. Here's a molecule of sucrose. Draw the monosaccharides that are produced when a molecule of sucrose is broken down. Now, this is a bit long, but you basically just want to copy the 2 molecules out that are drawn above and split them in half where the glycosidic bond is, and you need to add in a water molecule. So we want to just add on 1 O and 2 H's. So here I am drawing out the first molecule here, which is a hexose sugar made of 6 carbons. And of course, this is glucose that I'm drawing here. And this is our lovely glucose molecule. And we're just drawing the 6th carbon, CH2OH. Okay, so that's glucose. Now notice I've put an OH group. So I've returned a hydroxide group, which was missing from that one. Now all I want to do is redraw this one here and attach an H, because I've already stuck an OH there. We've got 1 more H of the water to attach. So I'm going to redraw this 5 carbon sugar. Now this is a pentose sugar, and this is known as fructose. You've probably heard of that before. Fructose, well, sucrose is a disaccharide. It's made of fructose and glucose, and this is the fructose. I just dodged up that bond. Let me just sort that bond out. And all I need to do here is just add an H on, and that will mean I have added back the water molecule. Super duper duper. And there you go. I've circled in blue where I've added the two H's and the O back on. And that's it. Simple as. The name of the two monosaccharides I already mentioned. One is glucose and one is fructose. There we go. Okay, moving on. We're getting near the end now. Just a couple more questions. Monosaccharides join to form disaccharides and polysaccharides. Compare and contrast. Oh, not again. Compare and contrast the structure of a disaccharide with glycogen. I've already done this with, was it maltose and glycogen we did earlier? So compare means the same. Contrast means different. Pause the video. Have a go. You should be able to get this by now. All right, so don't spell disaccharide like that, but a disaccharide is made of two monomers, two glucose monosaccharides. If we're talking about maltose, we could be talking about sucrose as a disaccharide, in which case it's made of glucose and fructose, or lactose, which is glucose and galactose. So it does depend which disaccharide you're talking about. I'm just going to mention maltose here. It's made of two glucose monomers. Both contain glycosidic bonds. So both glycogen and disaccharides contain glycosidic bonds. Both contain the elements carbon, hydrogen, and oxygen. So there's quite a few similarities there. I'd say the best ones to go there for your 2 marks out of the 4 for similarities are they both contain glycosidic bonds and both contain carbon, hydrogen, and oxygen. However, here's our first contrast. Glycogen has 1 to 4 and 1 to 6 glycosidic bonds as it's highly branched, whereas disaccharides, of course, are only made of 2 sugars with 1 bond in between, so they are not branched. So disaccharides only contain 1 glycosidic bond that is not branched. And now that glycosidic bond can be a 1 to 4 glycosidic bond, but as we saw with trehalose, which is a disaccharide, it could be a 1 to 1 glycosidic bond, but certainly it's not going to be have 2 different types of bonds. And there we go. Picking up the marks. Right, we're into the final question now. So last question, and then we're going to finish. Make sure you share, like, and subscribe. Mucopolysaccharides are complex molecules found in the human body. Mucopolysaccharides, what a word, can be broken down by enzymes. Describe how an enzyme would break the polysaccharide component of the mucopolysaccharide down. Okay, biology loves to throw these things off, but really they're just saying, how would it break a polysaccharide down? So what type of reaction would the enzyme do to break down a polysaccharide? The whole muco bit is just to throw you off. So all you need to be thinking about, what type of reaction breaks bonds? Is it condensation? Nah, it's not. It's hydrolysis. So the enzymes must do a hydrolysis reaction in which they break a glycosidic bond and release and use a molecule of water, not release. That would be condensation. I mean, you could, if this was more about enzymes, you could talk about the active site of the enzyme being complementary to the glycosidic bond in a specific polysaccharide, but we're not going to get into that here.
You're still here?
There we go. And that's your lot.
It's over.